It's a real privilege and honor to be here, and I've found this symposium absolutely fascinating. I wish I came to lectures at other times about the first 10 to the minus 22nd of the universe. It's really been a fantastic time. I've actually changed the topic of my lecture for a rather disappointing reason, and I'll show you that in a second. So, this is the reason. So this was the headline in the New York Times last week. And actually, in my view, this drug was the first test in experimental medicine of the amyloid, the first pure test of the experimental test of the amyloid hypothesis. And as you can see, it's a failure, and that's something we have to grapple with. So since the middle of last week, we're dealing with a slightly different future than the future that we might have imagined. And so I want to talk about why we developed the future we had, and what this finding really means for the new future that we're going to have. So I think I can't just sail on into the blue yonder, pretending in this post-truth era that this did not happen. So, in the interests of some truth, I'm going to try and deal with this issue. So, this is the pathology of Alzheimer's disease. There's amyloid. You're looking at something about a tenth of a millimeter across. That horrible black lump is amyloid, made up largely of the amyloid peptide. The wispy black stuff is tau tangle filaments, the tau protein. So this is the histopathology, and besides this histopathology, you've got massive neuronal loss. And this is what's afflicting about 3 million people here in the United States directly, and about 1 million people in the UK directly. So it's something we have to deal with as a society. Now, of course, faced with this pathology, scientists group themselves into the groups that you might expect. Those that said amyloid came first, tau second, and cell loss third. Those that said tau first, then amyloid, and then cell loss. And those that said cell loss was the important thing, and the amyloid and tangles were just the gravestones. And so, as a geneticist, I saw our role really as refereeing between those ideas. With that, we and others have studied the genetics, and this really summarizes the genetics. Those people with Down syndrome get early onset. People with Down syndrome, people with 3 copies of chromosome 21, get Alzheimer's disease. The amyloid gene is on chromosome 21, so we can be fairly sure for this and other reasons that overproduction of amyloid causes Alzheimer's disease. I'm going to show you in a second the first family with an amyloid mutation which caused Alzheimer's disease. The other gene was found by Peter Hislop. It's the presenilin genes, and they both are the enzymes responsible for yielding amyloid from the amyloid precursor protein. And incidentally, there is also a mutation found in Iceland which reduces the production of amyloid over your lifetime, and that reduces your incidence of Alzheimer's disease to about 1/3 or 1/4 of the general population rate. So all of these rare causes and this rare exception which reduces risk all relate directly to amyloid. However, although this is pretty impressive for the amyloid hypothesis, it only deals with perhaps 0.5% of the total number of cases of Alzheimer's disease. And there's been an enormous amount of argument about whether it's really justifiable to extrapolate from the rare to the general. Over the last few years, we and others have looked very carefully at the genetics of late-onset Alzheimer's disease. When we look at the genetics of late-onset Alzheimer's disease, there's the original finding of ApoE made by Alan Roses in the 1990s, and in the last few years we've found many other brain cholesterol genes are involved in Alzheimer's disease. The other The other classes have been proteins involved in APP processing, so of course directly related to the amyloid hypothesis, and then also microglial brain inflammation involved in the response to amyloid deposition. And I listened to the last talk about inflammation being a general risk, and that is certainly the case here with regard to Alzheimer's disease, that many of the diseases involved in late onset Alzheimer's disease are indeed genes involved in inflammation, and they respond directly to amyloid deposition. So this is the first family we found with amyloid mutations in 1991, and of course they had the tangles and the cell loss. This was the family tree with the grandfather affected, 2 sons affected, and then 5 out of 10 children on the left-hand side and 1 out of 3 on the right-hand side. The amyloid gene showed co-inheritance, and we sequenced the amyloid gene in this family. I will mention just briefly with this slide that this family was brought to us by the woman I'll show you in the last slide, who is a remarkable woman, and I'll talk about her at the very end. So this really allowed us to say that we believed in the amyloid hypothesis. We started to collect families with a different pathology. We started to collect families who only had tau mutations. These are quite rare, and it's nearly always a genetic disease. And when we looked for the gene for this disease, we found it was in fact the tau gene. So what we showed here was that if you have mutations in the tau genes, you got tangle pathology and cell death. And that really allowed us to make mice with tangles by putting tau mutations into mice and developing pathology. We crossed these mice with amyloid mice, and the important thing here is that what happened when we did this cross is that in the tau-only mice, we got pathology in the subcortical regions, but when we crossed the tangle mice with the plaque mice, with the amyloid mice, we got tangles in the cortex. And so this experimentally shows that amyloid is upstream of tau. So we now have created the amyloid hypothesis in an experimental system, and this was the drawing we drew upon getting these data. Now, I will say that when we drew this drawing, we thought that these irritating question marks would be sorted out within a year or two. But this drawing is now 16 years old, and we still have not worked out really in detail any of the question marks. So we still believe this general framework is true, but it's extremely disappointing that we still have these question marks. So we still don't really understand the direct relationships between the pathologies. We still don't directly understand. I assured the person that I knew what I was doing. It was a mistake. Here we have it. We still don't know. This question mark in particular, we still don't understand. And this is a serious problem, which I hope we can start to address. One thing that has become clear, and it's a good thing to talk about this here in San Francisco, is that all of these pathologies become self-sustaining, prion-like. So, for example, amyloid pathology spreads throughout the brain, and tangle pathology spreads throughout the brain. There's very nice data from Karen Duff, Brad Hyman, and Michel Gerdert showing that once initiated, tangle pathology develops a life of its own and spreads throughout neuronal networks. And we have to now start to consider what that means for treatment strategies if you initiate tau pathology and it is then self-sustaining. And that, I think, is key to part of the problem. In the last 3 years, there has been considerable progress in the imaging of these pathologies in the human brain. Previously, what we've had to do is rely on autopsies for our understanding of the disease, and this, of course, has not given us any idea of the sequence of pathologies. Amyloid pathology has been around for, amyloid imaging has been around now for nearly 15 years. And amyloid pathology clearly develops very early, and I'll show you a time frame in a second. And it's not been clear before what the relationship between the development of amyloid pathology and the development of tau pathology has been. But with the development of tau imaging agents in the last 2 or 3 years, we have started to become able to understand this. And what you have here shown on the left is tau imaging in a very young person on the extreme left, and then a person in their 40s here, and they have some evidence of abnormal staining, if you like, here. This does not seem to be tangles. Tangles are not found in this part of the brain. But here you see in the lower part of the brain just a beginning wisp of tau pathology. And this is in the absence of amyloid pathology. So you do get some tau pathology there in subcortical regions in 40 to 50-year-olds in the general population. If you have amyloid pathology, though, in the context of amyloid pathology, what you see is that tau pathology spreads into cortical regions. This is exactly the same as we had shown, as I showed you about 10 slides ago in the mice. So in the presence of amyloid pathology, the tau pathology is indeed exactly as in the mice, driven into the cortex, whereas in the absence of amyloid pathology, the tau pathology remains confined to subcortical regions. And so the mice, in fact, are a very good model of the progression of the pathology. But clearly, this is what we need to be able to prevent. Now, let's look at the time frame, because I think that this is the key to our problems. It's become clear because of these imaging technologies, and because we can follow these individuals in people with mutations, exactly what the sequence of events is. And this is the detection threshold. And here you have the time. This is, let's say this is 70 years old. This is when somebody comes to the clinic. They come to the clinic here because they're worried that they might at the beginning stages of Alzheimer's disease. When did the other things show? Well, the things that started 5 years before this, work actually from my colleague at UCL, Nick Fox, showed that 5 years before this, you started to see evidence for cortical atrophy. 10 years before this, you started to see evidence in the cerebrospinal fluid for abnormalities in tau. 15 years before this, you started to see amyloid PET changes. So the amyloid started to appear on the PET scans of individuals, and even before that, the CSF amyloids showed evidence that the amyloid was being deposited. So what we are now realizing is that when someone comes to clinic, they have already had the disease for 15 or 20 years. So all of the genetic and experimental data is still consistent with amyloid driving tau pathology. But there's two things we now have to take into account. The tangle pathology is self-sustaining and can be independent of the amyloid pathology. It occurs in the subcortical regions in the absence of amyloid pathology. And this process begins very early, very, very early. What does this mean for treatment? Well, the times that this trial was organized here, people just in the early stages of dementia. But their amyloid levels had already been hugely inflated by that time. In fact, they were maxed out, and the tangle pathology was already very, very widespread. And that means that simply it's an admittedly post hoc explanation of this failure is that the amyloid pathology was simply attacked too late in the disease process. And that if we want to attack the amyloid pathology, we are going to have to start looking at treating amyloid pathology at this stage in the disease process, rather in the same way that I took my statins this morning. Touch wood, I have not had a heart or attack or a stroke yet, but one would not take a statin for a heart attack or a stroke. In other words, the treatments have been aimed too late. How are we going to be able to detect people so early in the disease? Well, we can detect people in these mutation families quite straightforwardly, though with ethical considerations making it a little more complex. So in those cases, we can test therapies, and that is happening in two trials called the DIAN trial and the ADPI trial. What can we do, though, in the general population? Well, what we can do is use genetics to enrich people in the general population. And here I can see people at the ages of about 50 having, if you like, a genome-wide test using all of the genetic markers that we and others have found. And this allows a reasonably accurate prediction, an area under the curve of about 0.75, to determine people who are at high risk, who could then go on to have other more investigative investigations about their Alzheimer's status. To finish with this woman who really started the story, this is what she was quoted as saying in 1991 about this, and that she thought that we would not be able to help her, but we might be able to help her children. And she, unfortunately, well, I hope was realistic. Unfortunately, because she is in fact now sick with the disease, so tragically she fought effectively against it, but she is now in the middle stages of the disease. I hope that we will be able to treat her children. Thank you very much.
So do we have some questions?
Thank you for your talk. My younger brother was just diagnosed a year ago with early onset. He's 55, diagnosed at 54. But looking back, we were able to see now in retrospect almost 10 years of evidence that preceded that. I'm curious about if you have any thoughts on the mesenchymal stem cell trial that is just starting at Emory and UC Irvine in the United States, and whether you think that might provide some hope. I know it's impossible to predict, but I'm just curious how, in terms of your research, how getting an infusion of mesenchymal stem cells might impact.
Yeah, I just think that I've not been a fan of stem cell therapies for Alzheimer's disease. I think much more for Parkinson's disease, as was mentioned this morning, because the cell loss is more circumscribed. I think it's very difficult to see how stem cells are going to work. They're great tools, and we're using them in our lab. We have cells from these mutation carriers, and we're growing spheroids and understanding the spread of pathology in them. So they're great tools, but I don't see them as treatment options. I'll just say, if you're from the San Francisco Bay Area, there's a great team here. Bruce Miller leads a great center on familial neurodegenerative diseases here in San Francisco.
Yes, Professor Hardy, I have a question about the name of the disease, the Alzheimer's disease. It's basically named after a physician, right? So I'm not quite satisfied with the nomenclature to describe the disease. It may now, I work in the cancer field. Now it's more and more realization about the heterogeneity of a cancer. So is there a way to better categorize Alzheimer's disease according to more like a molecular pathology, rather than the biochemistry ones?
Well, yes and no. Of course, you're right that the disease has many causes, and we have this argument in Parkinson's disease for exactly the same reason. I think that it's very useful for a patient to be told that they have Alzheimer's disease. It gives them a group to, I mean, we have to think about why we name diseases. It's not just for our own internal scientific discussion. I think that we scientists have to be aware that the nomenclature, like all human invented things, is imperfect. I'm happy with the name Alzheimer's disease. Of course, when we go in, for example, into this DIAN trial organized out of St. Louis, it's only people with presenilin encoded Alzheimer's disease, so in the way you suggest, which are eligible to be enrolled. So nomenclature is always imperfect. We have to take it with a sense of humor, but we have to remember that it's really for patients more than it's for us.
Let's thank our speaker again. Thank you, John.